Good morning, students. Today we will move ahead in Chapter 3, Human Reproduction, with the topics Gametogenesis. It includes Spermatogenesis and Oogenesis. What is meant by gametogenesis? So first of all, try to break the terms gamete and genesis. Gamete is the cell that is involved in the sexual reproduction, and genesis means to produce. Spermatogenesis, production of sperm, and oogenesis, production of ovum. The primary sex organs, testes in male and ovaries in female, as we have studied already, produce gametes, sperms and ovum respectively by the process called gametogenesis. That is, sperm और ovum के बनने की process को कहते हैं spermatogenesis and oogenesis respectively. In the testes, immature male germ cells, spermatogonia produce sperm by spermatogenesis. Spermatogenesis in human begins at puberty. Puberty is the age at which secondary sexual characters start developing and someone becomes reproductively active. The spermatogonia present in the inside wall of seminiferous tubule multiply by mitotic division and increase in number. Each spermatogonia is plural and gonium is singular, is a diploid and contains 46 chromosomes. Some of the spermatogonia, called primary spermatocytes, periodically undergo meiosis. Periodically means at different time intervals. A primary spermatocyte completes the first meiotic division, that is reduction division, leading to formation of two equal haploid cells called secondary spermatocytes. So, spermatogonia undergoes mitosis whilst primary spermatocyte undergoes meiosis. These secondary spermatocytes have only 23 chromosomes because they are made after meiosis. The secondary spermatocyte undergoes second meiotic division to produce 4 equal haploid spermatids. You know that in meiosis we have two divisions, meiosis 1 and meiosis 2. So after meiosis 2, 4 equal haploid spermatids are formed. The spermatids are then transformed into spermatozoa, that are sperms, by the process called spermiogenesis. So what happened exactly is spermatogonia converted into primary spermatocyte, into secondary spermatocyte, into spermatid, and finally to the sperms. After spermiogenesis, sperm head becomes embedded in the Sertoli cells. You know that Sertoli cells were the cells meant for the nutrition and then are finally released from the seminiferous tubules by the process of spermiation. Spermatogenesis starts at the age of puberty due to significant increase in the secretion of gonadotropin releasing hormones. In this diagram, it's clearly evident that spermatogonium give rise to primary spermatocyte, then to secondary spermatocyte, and then to spermatozoas. And the in-between cells are known as Sertoli cells. Gonadotropin releasing hormone is known as GnRH. It is hormone released by hypothalamus. If you can recall, we have already studied this hormone in 11th standard. The increased level of GnRH then acts at the anterior pituitary and stimulates secretion of two gonadotropins. Underline the two gonadotropins, luteinizing hormone and follicle stimulating hormone, that is LH and FSH. LH act at the Leydig cells. Do you remember Leydig cells, interstitial cells, and stimulate synthesis and secretion of androgen. Androgen in turn stimulate the process of spermatogenesis. FSH act on Sertoli cells and causes them to secrete some factors that are required for spermiogenesis. Spermiogenesis, as we have discussed, it is the process of formation of sperm from spermatids. That is, structure of a sperm is getting ready in this process. So let us understand the structure of sperm. So in the microscopic structure, a sperm is composed of head, neck, middle piece and tail. A plasma membrane envelopes the whole body of the sperm. The sperm head contains an elongated haploid nucleus, anterior portion of which is covered by a cap-like structure, a chromosome. Mark the four parts: head, neck, middle piece, tail, haploid nucleus and cap called a chromosome. We can see these structures clearly in this diagram. Whole sperm is covered with the plasma membrane. The cap-like structure is a chromosome. Nucleus contains chromosomal material which is haploid, that is 23 chromosomes only. The neck part joins head with the middle piece. Middle piece consists of highly coiled mitochondria, energy source for swimming, and the tail. The acrosome is filled with enzymes that help the fertilization of the ovum. Middle piece consists numerous mitochondria which produce energy for the movement of tail that facilitate the sperm motility essential for fertilization. What it means is, एक्रोसोम में ऐसे एंजाइम्स होते हैं जो ओवम को फर्टिलाइज करने में स्पर्म की हेल्प करते हैं. वैसे ही मिडल पीस में बहुत सारे माइटोकॉन्ड्रिया होते हैं और जैसे कि हम जानते हैं कि माइटोकॉन्ड्रिया का काम क्या है, एनर्जी प्रोड्यूस करना. सो मिडल पीस जो है वो एनर्जी प्रोड्यूस करता है स्पर्म के लिए ताकि स्पर्म इजीली मूव कर सके, जो कि फर्टिलाइजेशन के लिए बहुत ज़रूरी है. Human male ejaculates about 200 to 300 million sperms during coitus, of which for normal fertility at least 60% sperms must have normal shape and size, out of which 40% must have vigorous motility to reach to the ovum. Sperms released from the seminiferous ducts are transported by the accessory ducts. Secretion of epididymis, vas deferens, seminal vesicle and prostate are essential for maturation and motility of sperms. The seminal plasma along with the sperm constitute semen. The functions of male accessory ducts and male accessory glands are maintained by androgens. Next gametogenesis is oogenesis, that is the process by which ovum is produced. It doesn't start at puberty. It's somewhat different from the spermatogenesis. Let us mark the differences between them and also study the oogenesis. The process of formation of mature female gamete is called oogenesis, which is markedly different from the spermatogenesis. It is initiated during embryonic development stage. Mark the difference. It initiates at embryonic developmental stage when a couple of million gamete mother cells, that is oogonia. There in spermatogenesis it was spermatogonia, here it is oogonia. They are formed in the fetal stage, fetal ovary. No more oogonia are formed and added after birth. These cells start division and enter into the prophase 1 of meiotic division and get temporarily arrested at this stage. So after starting meiosis, they get at the rest. These are called primary oocytes. Each primary oocyte is surrounded by a layer of granulosa cells and is called primary follicle. A large number of these follicles degenerate during the phase from birth to puberty. Therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. The primary follicles get surrounded by more layers of granulosa cells and a new theca and are called secondary follicles. The secondary follicle soon transforms into a tertiary follicle, which is characterized by fluid-filled cavity called antrum. The theca layer is organized into an inner theca interna and an outer theca externa. This is the structure of secondary follicle. जिसके अंदर जो कैविटी है वो फ्लूइड से फिल्ड है, जिसे एंट्रम कहते हैं। जो उसकी लेयर्स हैं वो इंटरनल है और एक्सटर्नल है। इंटरनल को बोलते हैं थीका इंटरना एंड एक्सटर्नल को बोलते हैं थीका एक्सटर्ना। It is important to draw your attention that it is at this stage the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division, means it comes out of the rest period. जो resting stage में वो चला गया था उससे बाहर आ चुका है, वो बहुत बड़ा होने के बाद division के लिए ready है. It is an unequal division resulting in formation of large haploid secondary oocyte and a tiny first polar body. दो सेल्स बनते हैं, एक सेकेंडरी oocyte और दूसरा टाइनी फर्स्ट पोलर बॉडी. सेकेंडरी oocyte retains bulk of the nutrient rich cytoplasm of primary oocyte. Thus the first polar body born out of first meiotic division divide further or degenerate. At present we are not certain about this. The tertiary follicle further changes into the mature follicle or graphene follicle. The secondary oocyte forms a new membrane called zona pellucida surrounding it. The graphene follicle now ruptures to release the secondary oocyte from the ovary by the process of ovulation. So ovulation क्या है? It is the process of releasing ovum from the ovary. Okay? Graphene follicle is ruptured now. Let us see the development of the follicles. Here in the ovary, blood vessels, primary follicles, very tiny follicles, they grow and become large. Tertiary follicles are showing antrum and cell. Then graphene follicle, a very large structure, ruptures, ovum is released. Then the empty follicles, empty graphene follicles are known as corpus luteum. Let us compare both spermatogenesis and oogenesis with the help of this chart. First of all, at puberty, the chromosome number of cell is 46. Here in female, it is in fetal life. Okay, so spermatogonia undergoes and mitosis and form primary spermatocyte. Here oogonia undergoes mitosis and forms primary oocyte. After this, the oocyte is arrested in female till birth. While primary oocyte undergoes first meiotic division, forms secondary spermatocyte, second meiotic division and spermatids. Here, after birth, childhood and puberty, when it becomes secondary oocyte by forming first polar body, the secondary oocyte undergoes reductive division and forms ovum and secondary polar body is produced. It occurs in adult reproductive life. Here, whole spermatogenesis occurs in adult reproductive life. Secondary spermatocyte as well as secondary oocyte are always haploid. Spermatids are haploid, ovum is haploid. Spermatids undergo differentiation, that is a spermiogenesis, and forms spermatozoa that are functional sperms. Release of sperms is known as spermiation, while release of ovum is known as ovulation. Next is menstrual cycle. We have already read that all the primates undergo cyclical changes, and we human belongs to the class where menstrual cycle is observed in female as the cyclical changes in the placental mammals. In menstrual cycle, the reproductive cycle of female primates like monkey, ape and human being is menstrual cycle. The first menstruation begins at puberty and it is known as menarche. So start or begin of the menstrual cycle is remarked as menarche. In human female, menstruation is repeated at an average interval of about 28 to 29 days. The cycle of events starting from one menstruation to next is known as menstrual cycle. One ovum is released, that is ovulation occurs during the middle of each menstrual cycle. The major events of menstrual cycle are shown in figure. The cycle starts with the menstrual phase when menstrual flow occurs and it lasts for 3 to 5 days. What is this menstrual flow? It is result of the breakdown of the endometrial lining of the uterus. We have studied the lining of the uterus, perimetrium, endometrium, myometrium. So the endometrium breaks down and releases all the dead cells, tissues along with the blood. This is menstrual flow. Menstrual flow is blood vessels which forms liquid that comes out through vagina. Menstruation occurs only if the released ovum is not fertilized. Lack of menstruation may be indicative of pregnancy. However, it may also be caused due to some other underlying causes like stress, poor health, etc. The menstrual phase is followed by follicular phase. During this phase, the primary follicles in the ovary grow to become a fully mature Graafian follicle. Simultaneously, endometrium of uterus regenerates through proliferation. These changes in ovary and uterus are due to the hormones released by pituitary and ovary. We can see the hormones FSH and LH and their different levels at the different time periods. Here, a cyclic change is shown. The last one, the bottom one, is uterine changes, in which first 3 to 5 days are showing menstrual flow. Then it is getting regenerated. Then ovarian hormones estrogen and progesterone are shown. They simultaneously act on uterine recovery. Here, the luteinizing hormone and FSH simultaneously results in ovulation. Here, ovarian events are shown: development of follicles from primary follicle to Graafian follicle, and finally ovulation. After ovulation, yellow colored follicles are shown, that is corpus luteum. Okay, these are responsible for the ovarian hormones. Both LH and FSH attain a peak level in the middle of the cycle, that is about 14th day. Rapid secretion of LH leads to the maximum level during mid-cycle called LH surge. It induces rupture of Graafian follicle and release ovum, that is ovulation. So this way ovarian hormones and pituitary hormones both simultaneously act on ovary and uterus. Ovulation is followed by luteal phase, that is in which the remaining parts of follicle act as corpus luteum. It becomes secretory and it secretes large amount of progesterone, which is essential for the maintenance of endometrium. Such an endometrium is necessary for implantation of fertilized ovum that occurs in the pregnancy. But in absence of pregnancy, the endometrium breaks off. During pregnancy, all events of the menstrual cycle stop and there is no menstruation. In the absence of pregnancy, the corpus luteum degenerates. This causes disintegration of endometrium, leading to menstruation and marking the next cycle. In human being, menstrual cycle ceases around 50 years of age, that is termed as menopause. Mark the terms: starting was termed as menarche and ending is known as menopause. Cyclic menstruation is an indicator of normal reproductive phase that extends between menarche and menopause. So in this lecture we have covered these two topics, gametogenesis and menstrual cycle. In the further lectures we will be completing these three topics: fertilization, implantation, pregnancy and embryonic development, and parturition and lactation. Till then, keep studying.